Commensalism is plus 0, is it? One benefits and the other one is not benefited or harmed. Okay. Minus minus competition, plus 0 commensalism. The amount of nutrients such as carbon, nitrogen, phosphorus and calcium present in the soil at any given time is referred to as, don't look at the options, what do you call it? Standing state. What is standing crop? Biomass. In any particular geography, what is the amount of nutrient that is present in the soil is referred to as standing state. Standing crop is the entire biomass. What is a climax community? Give me an example of a climax community. Forest is a climax community. That is, you have all levels of organisms developed. You will have very minimal change in the species composition of that particular geographical region. That is a climax community. In that community, the species composition is more or less fixed. That is a climax community. What is a bare rock? A bare rock. What type of reference we can give to a bare rock? So life beginning on a bare rock. Whatever begins life on a bare rock, what do you call that? Succession, correct. Primary succession is the beginning of life. And succession eventually leads to climax community. What is the species that is the one which begins the succession in a particular region? Pioneer species. It is pioneer species. Okay, next one. Which of the following algae contains mannitol as reserve food material? Now see, we get a lot of questions related to algae. There are three types of algae. What are the three types? Green algae, brown algae and red algae. Each one of them have storage food. What is the storage food in green algae? Starch. What is the storage food in brown algae? Mannitol. What is the storage food in red algae? Floridian starch. Floridian starch. Some of the red algae are masters of producing agar. Who are those two fellows? Gelidium and Gracilaria. Gelidium and Gracilaria. Agar is produced. Some brown algae is specialized in producing anticoagulant algin. Alginates are produced by brown algae. Okay. So we have to remember this. So who produces mannitol as the reserve food material? Ectocarpus is a brown algae. Gracilaria is red algae. Volvox and Dulothrix are green algae. You have to remember, you can't help it, you have to remember the examples that are given in NCERT. In spite of interspecific competition in nature, interspecific meaning between species, which mechanism have competing species evolved for their survival? What do? Let's say you are a gangster, you have your own area of operation. A new gangster comes in. Okay. Gangster is a big word. You are a local rowdy collecting hafta from a particular area. One more person starts competing with you in the same area. What is the problem? Why do the shopkeepers pay you hafta? Hafta is referred to as protection money in English. Correct? I am not giving you career counseling. I am just giving you an example. Alright? To understand interspecific competition. So, I am a shopkeeper, I am giving you, let's say 1,000 rupees per week as protection money, hafta. What do I expect from you? That nobody troubles me. And whenever I am in trouble, you do the dirty job of beating up somebody or whatever it is. All the physical jobs you want, you ensure that you do it. Somebody else is also coming and asking me money for the same. Now what is happening? Conflict of interest. Conflict of interest. What will you try to do first? What will be your first attempt? Huh? What do you want to do? What do you want to do? Will you tell me not to give money, or will you go and tell that person not to collect the money? I am helpless. He is also coming with 10 people with all the items that can harm me. So what do you do? What is your first attempt? Come on, you. Clearly you are not fit for that profession. What do you do? You will go and warn that person. You will tell him, this is my area, don't set foot here. Okay? He will not listen to you. He is also powerful. You are also powerful. Fight goes on for a while. And eventually both of you realize that it is not possible for both of you to survive if you keep fighting. But both of you want to operate from the same area. Then, what is your arrangement?
Deal.
Huh?
Deal.
Deal. What is the deal?
We'll protect the owner together.
Huh?
We'll protect that person together.
Protect that person together. So I get double advantage at half cost. No.
This week you take money. After that I will take money next week.
Okay. Then I will start having favorites. He is better than this fellow. Do you want your protectees to start feeling that your competitor is better than you? You don't want your customers to have experience of another vendor also, no? So as a businessman, do you want to tell somebody, let's say if you have a provision store, will you tell your customer, this week you buy from my shop, next week you buy from that shop? And again for the third week you come back to my shop. Right? You don't have something that the customer is asking for. You know that it is available in the next shop. What do? Will you send the customer to buy it from the next shop? You will send your boy to get it from the next shop. You will give that item at cost price only. But you don't want the customer to go there. So will you tell, this week you collect, next week I will collect? What is the deal? Come on. Quickly, we are wasting too much time on this. Correct, you're right.
They will divide.
Divide the area. Those two streets you keep. These two streets are mine. Don't come here, I will not come there. Both are happy. No fight because a lot of resources are going in this fighting. It's okay, we will divide. Even if income becomes lesser, it's fine. We will increase our price. So we will balance our income, but at least we will not waste money on fighting. Same thing happens in nature. So look at the options. What is the right choice? Resource partitioning. You will partition the resources. If both of you are fighting for the same resource, you will partition the resource. Like some birds. You take the upper branches, I will take the lower branch. I will not come to the upper branch, you don't come to the lower branch. So whatever is in the lower branch is mine, whatever is in the upper branch is yours. Many people do this with God also, no? Resource partitioning. God, whatever I earn, first I will give it to you, you take whatever you want, whatever comes back is mine. Do it. Tell it to God. Whatever God wants, let him take. If you want to take completely, whatever remains, whatever falls down, I will take it with full humility. Resource partitioning with God also. Okay, next. Genera like Selaginella and Salvinia produce two kinds of spores. Such plants are known as heterospores. The two types of spores generally would be microspores and macrospores or megaspores. Site of perception of light in plants during photoperiodism. Repetition again. The previous year also had this. What is the site of perception of light in plants for photoperiodism? Leaves. Leaves are the places where the plant identifies light. Can you tell me, there are two theories there. Whether it is the duration of continuous light exposure or the duration of uninterrupted darkness. What is determining photoperiodism? Long day means there is shorter night exposure, shorter darkness, absence of light. Short day means there is longer darkness periods. So, which one is more influential? What do the plants want? If you interrupt the darkness with light exposure intermittently, will it still produce flowers? So photoperiodism is as much dependent on the continuous exposure to darkness or absence of light as it is to the exposure to light. Both are important. Plants follow different pathways in response to the environment or phases of life to form different kinds of structures. This ability is called plasticity. Plasticity is the ability to adapt to different conditions. Matching. Which one do you want to catch hold of first? Cristae. What is it? Infolding of mitochondria. A3 done. Statement 3. Option 3. Over, no? So simple this one. Maybe post-COVID paper. That is why it is easy.
Yes.
Cristae, infoldings of mitochondria, thylakoids, flattened membranous sacs and stroma of plastids, centromere, primary constriction in chromosomes, cisternae, disc-shaped sacs and Golgi apparatus. After you join your graduation and enroll for a gym, you will find one poster in every gym. There are gives you what? Virus-free plants. Micropropagation, somatic clones. Somaclones are what? Genetically identical plants. During the purification process for recombinant DNA technology, the addition of chilled ethanol precipitates out DNA. Complete the flow chart on the central dogma. Define the central dogma. Don't look at the flow chart or the sequence. Central dogma is what? Transfer of genetic information from DNA to RNA to proteins. DNA to RNA is referred to as transcription. RNA to protein is referred to as translation. So transcription, translation, proteins should be the sequence. The last three: transcription, translation, protein. Only statement C mentions it. A is what? DNA giving rise to DNA itself. Even if you don't understand what A is referring to, you can still do it with BCD. A is replication, DNA replication. Which of the following is not an application of PCR? Molecular diagnosis. Can we use PCR for molecular diagnosis? Yes. You take a sample of a cell, purify the DNA, amplify the DNA, and then do diagnosis. Gene amplification? Yes. Purification of isolated protein? No. PCR is done for amplifying DNA and study the DNA for whatever purpose you want to study it. It is not associated with proteins. So purification of isolated protein is wrong. What technique can you use for purifying proteins? Downstreaming is in the bioreactor. Chromatography. To separate proteins of different sizes, different molecular weights, you do chromatography. Chromatography is the technique. Plant hormone used to destroy weeds in a field is IAA, NAA, IBA. IAA, NA and MDA are not football teams. IA is indole acetic acid, NA is naphthalene acetic acid, IBA is indole butyric acid. All of these are oxygen analogs. They are all growth promoting chemicals. 2,4-D or 2,4-dichlorophenoxyacetic acid is a synthetic oxygen. 2,4-dichloroacetic acid is a synthetic oxygen used for destroying weeds. It can destroy broad-leaf weeds without damaging the crops. Again, matching question. Already it is a 200 question paper. And so many matching questions increases time consumption for many students. Those who are used to dealing with match the following questions will have advantage. Okay, which one do you want to catch hold of now? Cells with active cell division capacity. What are those cells? Meristematic tissue. A should be 2. Only one option. Over. Cross check with the other combinations. The option 1. B4. Tissue having all cells similar in structure and function. They are simple permanent tissue examples. Simple permanent tissue examples. Parenchyma, collenchyma, sclerenchyma. Arenchyma is what? Arenchyma. Arenchyma is, that is also parenchyma modified to have air inside that, inside air cavity. Tissues having different types of cells. Tissue having different types of cells. Vascular tissue, whether it is xylem or phloem, you have 4 different types of cells in the xylem and phloem. Dead cells with highly thickened walls and narrow lumen, sclerotes. Which of the following is not secondary metabolites in plants? Primary metabolites are essential for survival. Secondary metabolites are produced as a defense mechanism. Defense or for storage. Alright? Which of these are not secondary metabolites? Amino acids and glucose. Amino acids and glucose are required for their survival. Glucose for energy needs and amino acids are for building various plant organs. Which of the following statements is incorrect? Look at the statements. As soon as you get hold of a wrong statement, mark it and check for the other statements' correctness. During aerobic respiration, the role of oxygen is limited to the terminal stage. True. Why is oxygen required in the terminal stage? Oxygen is the terminal electron acceptor. Oxygen is the terminal electron acceptor. Okay. In the ETC, one molecule of NADH plus H plus gives rise to 2 ATP molecules and one FADH2 gives rise to 3 ATP molecules. Wrong. It is reverse. ATP is synthesized through complex 5. ATP is synthesized through complex 5. Correct. Complex 5 is also referred to as ATP synthase complex. ATP synthase complex is present in chloroplast also. True or false? ATP synthase complex is present in the chloroplast as well. True. What does it do? Same thing. The gradient for ATP synthase is outside to inside in chloroplast. The gradient for the flow of protons in chloroplast is from outside to inside. Is it true? From outside to inside? It is inside to outside. In mitochondria it is reverse. From the intermembrane space into the matrix, mitochondrial matrix. Oxidation reduction reactions produce a proton gradient in respiration. True. Can you tell me the sequence of electron carriers from donor to recipient? donor to acceptor in the electron transport chain of mitochondria, oxidative phosphorylation. Electron donor in complex 1, NADPH. Electron donor in complex 2, FADH. Both of them gives their electrons to before complex 2, between complex 1, 2 and 3, there is a mobile electron carrier. Who is that? Who is that? Plastoquinone, ubiquinone. Plastoquinone and plastocyanin are in the photophosphorylation. Here it is ubiquinone. Then from they transfer it to complex 3. Complex 3. From complex 3 it moves to complex 4 via cytochrome C. Cytochrome C. After complex 4 it moves to complex 5, which is complex 5 is ATP synthase, and the terminal electron acceptor is oxygen. See, in photophosphorylation, what is the sequence of cytochromes? There are other intermediate electron carriers also. At least remember this much: complex 1, complex 2, complex 3. Between them there is ubiquinone. Between complex 3 and complex 4 there is cytochrome C. Sorry. And there are various cytochromes associated in this entire chain. There you have to remember it as B, cytochrome B6F complex there in plants. Here it is BCA. Cytochrome B, cytochrome C, cytochrome A. BCA is a course also, no? Bachelor of Computer Application. It is an undergraduate course, BCA. Remember it as BCA. Again, 137 matching. Same repeat matching. Nitrococcus, Rhizobium, Thiobacillus, Nitrobacter. Which one do you want to catch hold of? Rhizobium. What does it do? Conversion of atmospheric nitrogen to ammonia. From school you are studying that Rhizobium is a nitrogen fixing bacteria. So Rhizobium, at least we should know. Rhizobium is participating in the conversion of atmospheric nitrogen to ammonia. A should be 4. B should be 4. Only one option. Even if you are not sure about the others, you still can get the answer. Nitrococcus, conversion of ammonia to nitrite. What do you call this? Ammonia to nitrite is called? Ammonia to nitrite is called nitrification. Thiobacillus is denitrification. Denitrification is conversion of ammonia to nitrogen. That is denitrification. Nitrite to ammonia is ammonification. So the opposite of nitrification is ammonification, not denitrification. Denitrification is reversing to nitrogen, atmospheric nitrogen. Nitrobacter, conversion of nitrite to nitrate. What is it? Nothing. As a process, we don't define anything. Okay, next one. This is problem. Correct? No? There are 4 floral formula given. One is very clear. 9 plus 1, 1 plus 2 plus 2. Corolla 1 plus 1, Corolla 1 plus 2 plus 2, and Andrecium 9 plus 1. That is Fabaceae. Fabaceae is 9 plus 1, and Fabaceae is what family? Legume, pea family. So let's see if we can catch hold of that and get the answer. Fabaceae is 9 plus 1 and you have the corolla arrangement as 1 plus 2 plus 2. 1 key petal, 2 standard petal and that arrangement. A should be 3. A3 only one option. Good, got saved. And Fabaceae is also zygomorphic. The other three families are actinomorphic. Fabaceae is zygomorphic, diadelphous, and 9 plus 1 arrangement. Identify the correct statement in capping. Capping happens in mRNA maturation for the purpose of what? Capping is what? Capping is methyl guanosine cap. At what end? At the 5' end for the purpose of protecting the code. Okay. In capping, methyl guanosine triphosphate is added to the 3' end of hnRNA. hnRNA is what? Heterogeneous nuclear RNA. Is it ready RNA for translation? It is still immature. It has to mature by capping and adding a tail. What tail do you add for RNA, mRNA? Poly A tail. Okay. So statement 1 is wrong. Statement 2, RNA polymerase binds with the Rho factor to terminate transcription in bacteria. Rho dependent termination in bacteria is common. Rho protein binds to RNA polymerase and it causes termination. That is correct. The coding strand in a transcription unit is copied to mRNA. Is it right? Is the coding strand copied to mRNA? Now this is a tricky statement. Coding strand is not used as the template. Coding strand is exactly the same, mRNA is exactly the same as the coding strand. Now how do we interpret this statement? Coding strand in a transcription unit is copied to mRNA. Do we have to look at it as mRNA has the same sequence as the coding strand? Photocopy? Or should we interpret it as coding strand is being used as a template to make the copy? How do you want to interpret that statement? If you interpret this statement as coding strand being used as the template for making the mRNA copy, then the statement becomes wrong. But if you look at it as mRNA is an exact copy of the coding strand, then it is correct. But we already have statement 2 which is correct without any ambiguity. So we have to make an assumption based on a little bit of convenience here. It's not a good statement. They have not given it properly. Split gene arrangement is characteristic of prokaryotes. What is split gene arrangement? Polycistronic mRNA. Split gene arrangement is exon, intron. The genes are interrupted with non-meaningful segments in between. Is that characteristic of prokaryotes? It is characteristic of eukaryotes. So statement 1, 3 and 4 are wrong. Statement 2 is right. Which of the following statements is correct? These are big questions actually. If you give 4 statements and you are told to identify how many of them are correct with multiple combinations. So it is like 4 questions in 1 question. Match the following, multiple questions in 1 question. You can increase the number of questions that you are asking by bringing in these kind of questions. This is at least 1 statement you are asking. Which of the following statement is correct? 1 statement is correct, other 3 are wrong. But if 2 are correct, 3 are correct, and you have options with combinations of those are time wasters. You will know. Even if you know it, it will waste time. So that is where you have to strategically act. Fusion of two cells is called karyogamy. Is it correct? Wrong. What is it called? Plasmogamy. Fusion of two cells can be considered as plasmogamy. In plasmogamy, is there karyogamy also involved? Is there karyogamy as well in plasmogamy? Or you are just fusing the two cytoplasm without nucleus? Plasmogamy is fusion of protoplasm. So protoplasm is exclusive of nucleus. Is protoplasm, see, in a cell, you have nucleus and cytoplasm. Within the nucleus, you have nucleoplasm. Correct? If you remove the nucleus and whatever is left out in the cell is what? Is it protoplasm? It is cytoplasm, no? Protoplasm is the entire constituent of the cell. Nucleus plus cytoplasm is protoplasm. Protoplast fusion is fusion of two cells, complete cells. Okay. So protoplasm is anyways. Fusion of two cells is not karyogamy. Karyogamy is fusion of two nuclei. Fusion of protoplasms between two motile and non-motile gametes is called plasmogamy. That is correct. See, plasmogamy will give rise to what? What does plasmogamy give rise to? Somatic hybrids. Plasmogamy gives rise to somatic hybrids, isn't it? You can take two somatic cells and bring about protoplast fusion, plasmogamy, and come up with a somatic hybrid. The hybrid will be viable with genetic material. You don't have nucleus, how will the hybrid be viable? So protoplast fusion involves the fusion of two cells complete. The nuclei and the cytoplasm also involved. The protoplasm fusion is happening there. Organisms that depend on living plants are called saprophytes. Is it true? False. What do you call it? Parasites. Saprophytes are that depend on decomposing matter. Some organisms can fix atmospheric nitrogen in specialized cells called sheath cells. Are the nitrogen fixing cells within certain organisms called sheath cells? Cyanobacteria have some specialized cells within them which are specialized for nitrogen fixation. What are those cells called? Heterocyst. Select the correct pair. Large colorless empty cells in the epidermis of grass leaves. What do you call those cells? Bulliform cells. What do? They help regulate water losses in the grasses. Bulliform cells, if it is turgid, the leaves will be open. If the bulliform cells are flaccid, the leaves will curl up. So it is not subsidiary cells. Okay. Subsidiary cells are found where? Where do you find subsidiary cells? Around the stomata. Subsidiary cells help the stomatal cells. They are not present in the epidermis. They are not the large empty cells. They are in the epidermis only, but not the large empty cells. In dicot leaves, vascular bundles are surrounded by large, thick-walled cells, and these cells are called conjunctive tissue. Is it right? Conjunctive tissue is found in roots, not in the vascular bundles of dicot leaves. Cells of medullary race that form part of the cambial ring. What do you call them? Interfascicular cambium. Loose parenchyma cells rupturing the epidermis and forming a lens-shaped opening in the bark. Spongy parenchyma. Is it spongy parenchyma? What do you call that? Parenchyma cells which will open the epidermis and form the gaps in the bark. What are they? Lenticels. What's happening?
They are calling themselves.
What is
What are these calling? Who? Okay, you should tell me, no? You are having internal conversations for some video recording, I suppose. Okay, interview. I will also get to see the interview. Okay. DNA fingerprinting involves identifying differences in some specific regions in DNA sequences called. See, DNA fingerprinting, what regions of the DNA do you identify? You identify VNTRs, variable number of tandem repeats. VNTRs, variable number of tandem repeats. And all the VNTRs in a DNA together is referred to as polymorphic DNA. It is referred to as polymorphic DNA. They are repetitive DNA only. It is a general term, not specifically applying to VNTR. Satellite DNA, what is satellite DNA? That is also repetitive DNA, but satellite DNA is not used for DNA fingerprinting. For DNA fingerprinting, VNTRs are studied. VNTRs are studied. In some members of which of the following pairs of families pollen grains retain their viability for months after release? Apparently it is Poaceae and Liliaceae. Poaceae is grasses and Liliaceae is Leguminosae. Leguminosae is legumes. Which of the following statements is incorrect? ATP and NADPH are synthesized during non-cyclic photophosphorylation. Is it correct or wrong? Correct. ATP synthesis happens at the end of the process, where you have ATP synthase associated with that, or the final terminal ATP synthesis happens during the process because of gradient that is produced. Electron transport is not associated with ATP synthase in this, but electron transport ends in the electron acceptor taking the electrons and becoming what? Who is the terminal electron acceptor in photophosphorylation? NADP. NADP is the terminal electron in photophosphorylation. If the electrons keep circulating within this pathway without going to the terminal electron acceptor, what will not be produced? NADPH will not be produced. But because the electron keeps flowing within this pathway, the gradient for hydrogen ions is produced. The flow of protons can still happen from the outside to the inside. So what will be produced? Inevitably, ATP will be produced. But NADPH cannot be produced because in cyclic photophosphorylation, the electron that is excited is not going out of this pathway to reach the acceptor, terminal electron acceptor. So NADPH will not be produced, but ATP is produced. The electron is excited in the first in the photosystem 2. It goes into the pathway, goes to photosystem 1 and all that, and it goes back, goes to plastocyanin, plastoquinone, then plastocyanin, photosystem 1 again goes back, ferredoxin, plastocyanin, photosystem 1, ferredoxin, plastocyanin, photosystem 1, keeps looping here. So the electrons excited from photosystem 1 will be replaced by the same electron which comes back. You understand? In cyclic photophosphorylation, do we have both the photosystems involved? Which photosystem is not involved? Photosystem 2 is not involved, and there is no NADPH production. These two you have to keep in mind and look at the options. Both ATP and NADPH are synthesized during non-cyclic photophosphorylation. Correct? Stroma lamellae have photosystem 1 only and lack NADP reductase. That is also true. Stroma lamellae has only photosystem 1 and there is no NADP reductase. Granular lamellae has both photosystem 1 and photosystem 2. Correct? Cyclic photophosphorylation involves both PS1 and PS2. Wrong. PS2 is not involved. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? See, there are 3 RNA polymerases. RNA polymerase 1, 2, and 3. RNA polymerase 1 is involved in the synthesis of larger fragments of ribosomal RNA. RNA polymerase 2 is involved in the synthesis of mRNA. RNA polymerase 3 is involved in the synthesis of transfer RNA and smaller fragments of ribosomal RNA. Now look at the options. Transcribes rRNAs 28S, 18S and 5.8S. They are larger fragments. So it is the role of RNA polymerase 1. Transcribes tRNA, 5S RNA and snRNA. What is snRNA? Small nuclear RNA, which is the correct option there. RNA polymerase 3. Transcribes precursor of mRNA. Who does that? RNA polymerase 2. Transcribes only sRNA, snRNA. In the exponential growth equation, nt is equal to n0 e to the power rt. E represents base of natural logarithms. In this, nt is what? N is what? N is population, t is at the time t, 0 is at the beginning. Nt is population at the end of time t, and 0 is population at the beginning. E is the natural logarithm, R is the intrinsic rate of growth, and D is the duration for which growth has. When do you see exponential growth in a population? When do you see exponential growth? When resources are unlimited. And when there is no competition. When there is no competition, there is exponential growth. With the weights, they will not even be nice to you. They will even scold you. You have to go and request them, please do. As competition increased, and they became very rich also because they are doing adulteration. People have to buy from them, no choice. So they are making a lot of money, and their wealth was growing exponentially. Competition began, and it became logistic growth for them. Now with competition, customer became the king. Customer became the king, and they will deliver it to your home. You can call them and inform them what you want. If you are busy, you can even send them a WhatsApp message. They will send it. Send a voice note, no problem, we will send it. Take it first, give money later, no problem. Okay. You want to buy something today? Okay, I'll give you EMI. I don't want to pay any interest on the loan. I will pay the interest. You please take it. Right. So, is competition bad? Competition is bad for people who are competing, but competition is good for the beneficiaries of that competition. Competition is bad for entrepreneurs mutually. But competition among entrepreneurs is good for the customers. What do? When you want to buy something, you will check both Flipkart and Amazon. Right? You are not loyal to Amazon or Flipkart. Your loyalty is only for the price. You will check which one is offering better discount. Even if it is 100 rupee discount on a 20,000 rupee product. Will you have loyalty to Amazon or Flipkart? Where do you order it from? You will order it from wherever there is 100 rupee lesser price. It is exclusively price dependent. So, value businesses have more loyalty. These are price businesses. Value business is what? Healthcare. You go to a particular doctor. I get calls. I have to go for my eye checkup. So I need 2 days leave. Why 2 days leave for eye checkup? My daughter has to see the same doctor. So to see the same doctor, 500 rupees for the eye checkup. Half a day to come here, half a day to overnight journey, 1 full day there, coming back. 3 days and 5 to 10 times more expenditure. Loyalty business. Alright. So My father's patients are also like that. It's nothing wrong, it's good. So you have to become somebody who can command that kind of trust. Trust-dependent businesses, you should always have a great sense of accountability and responsibility. It should always be value, not price. Price will always be there, you will always charge a price. But price should not determine your action, value, the outcome. What am I doing to them? Otherwise you will not survive as doctors. Nowadays, okay, it is possible to detect the mutated gene causing cancer by allowing a radioactive probe to hybridize its complementary DNA in a clone of cells, followed by detection using autoradiography. Because now see, you are suspecting cancer in a particular cell. You identify a fragment of the DNA. To which you hybridize. Fragment of the DNA in the normal cell. You know the sequence. You create a radioactive probe complementary to the DNA sequence of the normal cell. Now you denature the DNA, hybridize it with the radioactive probe linked DNA fragment. Then you observe for radioactivity. If the cell is normal, will it show radioactivity or will it not show radioactivity? It will show radioactivity because you have created the probe complementary to the normal DNA. But if it is a cancerous cell, it will not have the normal DNA, it would have what? It would have changed. So it will not complementarily bind to the cancerous cell DNA. If you don't find radioactivity in the cell in which you have treated with the radioactive DNA fragment as a probe, what does that indicate? It indicates that the cells that you are studying are probably cancerous. So it is diagnostic significance. Now look at the options. The mutated gene partially appears on a photographic film. Will it? No. The mutated gene completely and clearly appears on a photographic film. No. The mutated gene does not appear on a photographic film as the probe has no complementarity with it. That is correct. Probe has complementarity with the normal DNA, not to the mutated DNA. Did you finish giving your video bite?
She just informed us.
Oh, it was an information. Okay, right, next one. Matching again. Protein, unsaturated fatty acid, nucleic acid, polysaccharide. And there are bonds given. What? Protein? Peptide bond. Okay, A4. So option 2, 3 ruled out. Then? Unsaturated fatty acids. Unsaturated fatty acids will have carbon to carbon, double bond or triple bonds. Now it is double bond given, so it is fine. Okay, B should be 1. Option 1. Nucleic acids have phosphodiester bonds. Polysaccharides have glycosidic bonds. That's an easy one. Another matching question. Cell biology. S phase, G2 phase, quiescent phase, G1 phase. S phase, DNA replication, A4. Okay, option 1, 4 ruled out. Among 2, 3. Catch hold of next one. G, what? What do you want to choose? Quiescent stage, which is inactive phase, C2. Option 3. So 2B1, G2 phase is proteins are synthesized. G1 phase is interval between mitosis and DNA replication. Plasmid PBr322 has a PST1 restriction enzyme site within the gene for ampicillin resistance that confers ampicillin resistance. So the restriction site is within the ampicillin resistance gene. Alright, that is what you have to notice there. The restriction site is within the ampicillin resistance gene. If this enzyme is used for inserting a gene for beta-galactosidase production and the recombinant plasmid is inserted in E. coli strain, what do you notice? Now, if you put a foreign gene into the ampicillin resistance gene, what will happen to ampicillin resistance in that bacteria? It will lose ampicillin resistance. Correct? But what will activate this bacteria? What will activate the bacteria? The presence of ampicillin. Presence of ampicillin will activate the ampicillin resistance gene regulators. It will transcribe the ampicillin resistance gene, that segment. But in that segment you have removed a portion and inserted the gene of interest. So as long as ampicillin is present in lower doses, it will continue to transcribe it will continue to produce the ampicillin resistance gene segment in which you have your gene of interest. Look at the options. It will not be able to confer ampicillin resistance to the host cell. Is it right? It is correct. Because you have cut off the ampicillin resistance gene and put your gene of interest. The transformed cells will have the ability to resist ampicillin as well as produce beta-galactosidase. No. It will lead to lysis of the host cell. No lysis. It will be able to produce a novel protein with the dual ability. Nothing of that sort. Beta-galactosidase will continue to have what it is supposed to have as a property. Okay. Right? 50 questions done, 40 questions completed, 1.5 hours. Just 5 minutes extra. Take a 5-minute break. Alkaloids, codeine, toxin, abrin, lectins, concanavalin A, drugs, resin. Is codeine an alkaloid? Codeine is an alkaloid. Abrin is a toxin. Ricin is also a toxin, it is not a drug.
So,
Lecithin is concanavalin A. Come on.
yes,
Here. You're not waiting. I'll come. Okay, in the last class we discussed concanavalin A is a lectin. What are lectins? Carbohydrate binding proteins. 8 chromosomes, it is a diploid in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after the S phase? How many chromosomes after the S phase? 8 only. You will have double the content of DNA, but the chromosome number will not double, no. Each chromosome will have two chromatids. Only when you separate them, they are individual chromosomes. So remember, chromosome is a unit of condensed chromatin. Each condensed chromatin is not a chromosome unless they are separate units. Okay? Every chromosome is a unit of condensed chromatin, but every condensed chromatin is not necessarily a chromosome. How? Every chromosome is condensed chromatin, but every condensed chromatin is not necessarily chromosome. This is chromatin. Why does this dry up? The ink dries up because of cold. This is chromatin thread. You condense it. So this is chromosome. Every chromosome is condensed chromatin. Now we have two chromatin threads. Both the chromatin threads condense. Now how many condensed chromatin threads do we have in this? How many condensed chromatin threads do we have in this? 2 condensed chromatin threads we have. But is each of the condensed chromatin thread in this unit a chromosome? No. Each of them are chromatids. So we have one chromosome with two threads condensed. So it's like this, at the G1 phase, each chromosome will be like this. If it condenses, each chromatin is if it condenses, it becomes a chromosome. Here there is duplication of chromatin happening. Chromatin duplication happened. If it condenses, it will still condense into one chromosome, not two chromosomes. So that's a good question. It's an easy question, but this is a question where most of the students get it wrong. See, mitosis and meiosis is such a big problem for many people. But it is such a simple thing. All that you have to understand is how DNA has to be looked at in different contexts. That's all. Matching again. What do you want to catch hold of? Complex is of Xenophora. So C2. Option 1 and 2 ruled out. Option 3 and 4 we have to look at. C is 2. Then which one do you want to catch hold of here? Canal system is in Porifera B4. Option 3. Canal system is in Porifera. Metamerism is seen in Annelida. Is it true metamerism? Segmentation is seen in Annelida. Is it true segmentation?
Yes.
Metameric segmentation? Yes. Nidoblasts are coelentrates. What do they have? Specialized cells. Why are they called? Why are coelentrate are called nidoblasts? Why do they have? What are nidoblasts? They are stinging cells. Jellyfish. It is very painful now. Jellyfish looks very cute. You should not go and catch hold of it and say, oh, jellyfish. It will sting and painful. Okay. Contraceptives. Contraceptive methods. Matching again. Which one do you want to catch hold of? Vasectomy, tubectomy, easy. Vasectomy, removal of vas deferens, tubectomy, removal of fallopian tube. C2, D4. Only one option, which is second choice, second option. Vault is, where do you use vault? Vault is used for what? Vault is a barrier used by male or female? Female. Inserted into the vaginal part or the cervix part? Cervix, cervical vault. IUDs, phagocytosis of sperms in uterus. Is it true? Is it right? IUDs will promote phagocytosis of sperms in the uterus. Do IUDs phagocytose sperms? No. If adenine makes up 30% of DNA molecule, what will be the percentage of thymine, guanine and cytosine? See, if adenine is 30%, what should be 30%? Thymine should be 30%. So check for that option, which has thymine 30%. Option 3. Guanine and cytosine will be 20% each. What is the rule? This is basic equivalence rule. Chargaff's rule of basic equivalence. Which of the following RNAs is not required for the synthesis of proteins? siRNA. What is siRNA? What is siRNA? What is siRNA? Small interfering RNA. This siRNA is responsible for dash. siRNA is responsible for what? Something where? Correct, tell me. Introns? No. siRNA is responsible for something in tobacco plants. In tobacco plants, you use siRNA to bring about some change in the tobacco plant where some insects will not be able to damage this. Gene interference. In gene interference, you use siRNA or small interfering RNA. siRNA or small interfering RNA is the one which is used in gene interference. What is gene interference? You use a segment of the RNA which will block the expression of certain genes. And those genes, when it is blocked, will lead to destruction of the organism that you want to destroy. It is a biocontrol mechanism, gene interference. Not biocontrol mechanism, genetic regulatory control mechanism. Which one of the following is an example of a hormone releasing IUD? CUT, LNC20, CU7, multiload 375. Multiload 375 sounds like some gun. AK-47. Like we have a Indian army added a new tank, multiload 375. Battle tank. AK-47 is gun, no? Smaller name. Multiload 375. Should be.
Those who are doing the interviews.
Audio please.
Also those who will be doing the interviews and that's probably.
Okay, whoever is supposed to give interviews, not interviews, it's just monologue. You have to speak whatever comes to your mind. They give you mic but speak loudly. In front of camera, all of you will speak. What you all have practiced, what you all are good at, is very useful to irritate teachers. Teachers don't like blank expressions. If you don't like some teacher, you want to irritate the teacher. Is it recording?
Yes.
Okay, good. It will be useful for other students also. If you want to irritate teachers, you just have to, there is a body language for that. Like this. You should not open your mouth too much. It should just be like this. And you should relax your shoulder a little bit. Hold your hands. It should be like a ball of soft soil. And if they ask you something, you should be like, you should blink more. Can't do anything. Your body language will not encourage the teacher to punish you. But it will not let them be in peace. It's kind of. Okay. We should have some form of defense, no? You cannot always be oppressed. The oppressor also should feel some pain. Oppressors feel happiness in oppressing you. But you should also develop non-dangerous ways of making the oppressors feel uncomfortable. In this case, I am talking about the teachers. Succus entericis is referred to as out of syllabus question, intestinal juice. Chronic autoimmune disorder affecting neuromuscular junctions. Don't look at the options. Chronic autoimmune disorder affecting neuromuscular junctions leading to fatigue, weakness and paralysis of skeletal muscles is myasthenia gravis. Autoimmunity against acetylcholine, neurotransmitter acetylcholine. So in myasthenia gravis, the smaller skeletal muscles will get affected faster, like your eyelids, drooping eyelids. The patient will not be able to open the eyelids properly. It will be drooping, half sleepy eyes. Arthritis is inflammation of the joints. Muscular dystrophy is, what is muscular dystrophy? It's a genetic disorder. The most severe form of muscular dystrophy is DMD, Duchenne's muscular dystrophy, which is X-linked recessive disorder. Gout is accumulation of uric acid in joints. if you in gout you give protein restricted diet. Uric acid is a byproduct of protein metabolism. So in gout you give protein restricted diet, allowing the liver to recover and develop better abilities of managing amino acids. With regard to insulin, choose the correct option. Choose the correct option. So this is what I was telling. There are 4 statements given. Options are combination of statements with correct statements. Now be very clear. The moment you find something which is definitely right or definitely wrong. See, the question is asking you to choose correct options, correct statements. So if you find something which is definitely wrong statement, you have to eliminate the options which has that particular statement. If you find something which is definitely right, you need to eliminate the options which do not have that thing. Okay, now let us see. C-peptide is not present in mature insulin. It is true. So the right option should have A also included. The one without A should be eliminated. What do you eliminate now? 1, 2, you eliminate. Now look at 3 and 4. A and D are common to 3 and 4. Check for C, statement C. Proinsulin has C-peptide, which is true. Go for the right answer. You see that? So you have to act quickly on these kind of questions. You have to act strategically on these kind of questions. Statement B also, let us see. The insulin produced by recombinant DNA technology has C-peptide, which is wrong. What is the bonding between the two peptides in the 2 strands of protein in insulin? A and B strands? Disulfide bonds. That's all. Related to insulin, that's all you will get. And it is produced by recombinant DNA technology. Which organism is used for producing insulin? E. coli is used for that. Who produced it first? Eli Lilly. What do you call recombinant insulin? Human insulin. That's all. Nothing more than this can be a part of any question related to insulin. There can be, function and all can be, but this much is sufficient. Which one of the following belongs to the family Muscidae? Housefly. Musca, no? Housefly, Musca domestica. Which is the only enzyme that has the capability to catalyze initiation, elongation and termination in the process of transcription in prokaryotes? DNA dependent RNA polymerase. This is the 3rd question we are talking about in the last 2 days. Previous paper also had this. Correct? Well, the question was what in the previous paper? It was which one brings about unwinding of DNA during transcription? RNA polymerase. But because it is acting on the DNA, it is DNA dependent RNA polymerase. Among 1, 2, 3, RNA polymerase 1, 2, 3, which one is this? Which RNA polymerase is associated with transcription?
This only one.
2. RNA polymerase 2. RNA polymerase 1. See, in this paper only we discussed, no? RNA polymerase 1, 2, 3. RNA polymerase 1 is associated with large rRNA production, large components of rRNA production. 3 is associated with tRNA and smaller fragments of rRNA. RNA polymerase 2 is associated with transcription. Right? So it is RNA polymerase 2. Receptors for sperm binding in mammals are present on corona radiata, vitelline membrane, perivitelline space, zona pellucida. Are all 4 related or not? All 4 are related to ovum. But in mammals, do we have vitelline membrane? Vitelline membrane is present in amphibians, frog. These are all part of the ovum. From inside to outside, what all do you find? If you are coming from inside of the ovum to the outside, what all do you encounter? What do you encounter first? What is the barrier that you encounter first which you have to cross? No? Not of these four. I'm just talking about the ovum. You are coming from, you are inside the ovum now. You have to get out of this. What do you have to break? What do you have to break open first? If you have to come out of any cell, what do you have to cross first? Cell membrane, no? The ovum membrane, cell membrane. After outside of the cell membrane, what do you have? What do you have? Zona pellucida. Zona pellucida. Outside of that, you have cells. What are those cells? Corona radiatum. Zona pellucida is a glycoprotein layer. Now, in the sperm, what is it that will attach to the? What is it that will break open the zona pellucida? The acrosomal head, the acrosomal cap. Inside that, on the head, there is an acrosome, inside which there are hydrolytic enzymes. What are those hydrolytic enzymes? Coronalysin, zonalysin. These are the hydrolytic enzymes which will puncture this. Zona pellucida is where it will go and attach and release the hormone enzyme and break open the zona pellucida. So the receptor for sperm binding in mammals are present on zona pellucida. Acrosomal contents, the contents of the acrosome are produced by which organelle of the sperm? Contents of the acrosome are produced by which organelle of the sperm? Mitochondria. Think about what it is required for and correlate it to what can produce those things. What is there in the acrosome? What is there in the acrosome? Hydrolytic enzymes. Hydrolytic enzymes. So who produces hydrolytic enzymes? Huh? Lysosome. Lysosome is a product. Who is the manufacturer? Golgi complex. So Golgi complex produces these hydrolytic enzymes. So can we say acrosome in the sperm is like Golgi complex of the cell? The same hydrolytic enzymes are produced, not the Golgi complex, the lysosome of the cell. Hydrolytic enzymes are produced and packaged and placed in the head to break it open. Okay. Next. Centriole undergoes duplication during, when does the centriole duplicate? During the S phase. The other proteins, protein replication, protein replication, protein synthesis, G2, protein synthesis, G2. G1 is growth basically, and for production of enzymes necessary for DNA replication. Which one of the following organisms bears hollow and pneumatic long bones? Neophron is vulture. Neophron is vulture. Marcopus is a kangaroo. Ornithorhynchus is platypus. Hemidactylus. Hemidactylus. Reptile. Hormone erythropoietin, which stimulates RBC formation, is produced by, again repetition, we have discussed this hormone previously also. Erythropoietin is produced by juxtaglomerular cells of the kidney. In response to what? What is the stimulus for erythropoietin production? What does it identify? Does it look for the RBC density? What? Oxygen. If the blood that is carrying, if the blood that is going to the juxtaglomerular, going around the juxtaglomerular apparatus is constantly having low levels of oxygen, that means the oxygen concentration of the blood is low. Can be because of various reasons, but this fellow will identify it as decreased RBC production, and it will produce erythropoietin, which will go and stimulate the bone marrow to produce RBCs. Okay. What do you call the condition in which there is reduced oxygenation of blood? Reduced oxygen in the blood. Reduced oxygen. Hypoxia. hypoxia. Okay. What is asphyxia? ASPHYXIA, asphyxia. In a postmortem report, if it is mentioned, death due to tracheal compression leading to asphyxia. What is asphyxia? So what is this postmortem report talking about? What can be the cause of death there with that statement? Tracheal constriction leading to asphyxiation is the cause of death. Huh? How did the person die? Hanging. Tracheal compression. Trachea has been compressed. It can be either hanging or somebody has killed by tracheal compression. There will be other evidences also. There will be other evidences also to indicate whether it was suicidal death or homicidal death. Forensics is very nice. I was very interested in forensic science during MBBS. Once there was a exhumation request that was placed by a police station to our medical college. What is exhumation? Exhumation is you dig out a buried body and study it for, do a forensic analysis of the body. Our HOD was very fond of me then, head of the Department of Forensic Medicine. He was fond of me because I would be spending a lot of time in the mortuary, in the autopsy, in the post-mortem hall. So he asked me, do you want to come for exhumation? I was super excited. Yes, sir. We went there, it was about 15 kilometers drive. In Bangalore, 15 kilometers will take about 1 hour. So for 1 hour we went in our medical college van. Police also joined in. We went to a particular place. They already had, under the supervision of police, they had dug up the body. The person had died about 2 weeks ago. It was almost a molten body. Half a kilometer you can get the stench. Because it was already open, no? Exhumed. Our HOD is going happily with this mask on. He is going, picking up samples, collecting samples and all. I felt not to do forensic medicine further. So it is exciting to understand cause of death. There is mystery, no? It's like an investigation. But I felt exhumation is also a part of that. So, okay. Why did that come? Hypoxia, anoxia, asphyxia. Asphyxia, you have to remember, compression. Obstruction to airflow is asphyxia. Choking, correct. Choking is asphyxia. Choking is also asphyxia. Next, matching. Physalia, limulus, ankylostoma, pinctada. What is Physalia? Portuguese man of war. Limulus, I know it is crab, king crab. Limulus is king crab, which is the living fossil. Ankylostoma duodenale is hookworm. Hookworm, which causes hookworm disease, one of the major causes of anemia also. So that's all, match it. Pinctada is pearl oyster. 3, 2, 3, 4, 1. During the process of gene amplification, see, you know the mechanism of pearl formation in the pearl oyster? How does it produce pearl? There is this shell, there is a membrane. Pearl is a product of inflammatory reaction if you look at it, isn't it? There is some irritant that goes and settles down in that shell. Some irritant goes and settles down. So the pearl oyster, its gland starts producing some chemical, some substances. It starts secreting the substances to cover off that irritant. to not let that irritant escape. In that process, pearl is produced. Alright. So if pearl production is, and that increases the value of this pearl oyster. Value for humans, but it increases its risk of death also. No? If not for the pearl oyster producing pearl, why would humans be interested in that? Humans will be interested to eat,
Yes.
but catching hold of that will be not so easy, unlike fishes. But other ways also, if you look at it, it is a battle against some threat, which made the innate ability in the pearl oyster manifest into a product which is valuable. So nothing valuable comes out of comfort. Discomfort is the trigger for evolution or manifestation of abilities that can transform challenges into successes. So, how do we do it consciously? Do we have to go and live on the streets, study under street lights? No. Constantly you have to keep pushing yourself to the limit of your comfort zone, physically, intellectually, all that, by which you constantly develop yourself and align yourself to be ready to rise up to the occasion when a situation demands. Only when you are ready to face a challenge, the challenge will start appearing in front of you. If you are not ready for it, you will not be able to notice the challenge. And even if you notice it, if you are not ready for it, you will not be willing to take up the challenge. So pushing your comfort zone, waking up at 5 o'clock, going through this. Why, in spite of all of you asking me, many of you asking me repeatedly that we will sit in some other place, we will study in our room, we will study in the corridor, we will study in the library. Why I insist you sit here? Just pushing your comfort zone. Your body will tell you to get back to some zone of comfort. At that time you have to think what is right to be done, what is right. Do it. It will be uncomfortable, still do it. Keep doing it. Over a period of time, it starts becoming comfortable. So be ready to face challenges. Don't have the fancy ideas that I will work for 10 years and then retire. I ask youngsters, what is your dream? Sir, I want to make so much money so fast that I can retire at the age of 40 and keep doing holiday. You will not for me. Pink Tada, you have to be so grateful for Pearl Oyster for teaching you the importance of struggle for survival. You have to fight. Fight for survival, not only just with the sense of regret or cursing God, why do you give me so many difficulties? No. You were given a challenge, I will grow myself so strong that I will overcome the challenge and transform that itself into an opportunity and progress. For most of the people, it is all just, there is a saying, you don't go to 6 and neither come to 3. Ar Kerala, more Kerala. It's in Kannada. Your life is between 3 and 6. You cannot cross 6, you don't come down below 3. That is the middle class mindset. You should get out of that mindset. Okay. During the process of gene amplification using PCR, it is a very high, if a very high temperature is not maintained in the beginning, which step will be affected first? Denaturation will be affected. Only when denaturation happens, annealing can happen and extension can happen. If denaturation only is not happening, no annealing or extension is possible. Which of the following statements wrongly represents the nature of smooth muscles? No striations, is it wrong? They don't have striations. Involuntary, they are involuntary. Communication among the cells is performed by intercalated discs. Is it right? It is wrong. Intercalated discs are present in which muscles? Cardiac muscles. Why?
Because they,
Why do you need intercalated discs?
because they perform as whole, as one thing.
Cardiac muscles function in an all or none context. That means a signal reaching a particular muscle at one point should reach all the muscles in that group at that very instant. So you need to have extremely fast communication between the muscle fibers. So cardiac muscles have intercalated discs, and that is the wrong statement about smooth muscles. Smooth muscles are present in the wall of blood vessels, that is also correct. Organelles that are included in the endomembrane system. Don't look at the options. Endomembrane system is a continuous system of membranes associated with transformation of genetic code into products till the end of it. So tell me who are all involved. Is nucleus involved as a part of in the endomembrane system? Is nucleus a part of the endomembrane system? It is. Nucleus is a part of the endomembrane system because the blueprint for the product is coming from the nucleus only. Then connected to that is the endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. Both are associated with it. Proteins are produced, proteins are modified. Lipids are produced in the smooth endoplasmic reticulum. Then where these products go? They go into the Golgi complex. They go into the Golgi complex where they are packaged or further modified and the final product is released. Released directly into the cytoplasm or packaged into vesicles to be delivered outside. So do we consider vesicles as a part of the endomembrane system? Should we consider vesicles as a part of the endomembrane system? Vesicles or vacuoles? Vacuoles are big vesicles, pouches. Should these vacuoles be considered as a part of the endomembrane system? Yes or no? Yes. Should we consider lysosome as a part of the endomembrane system? Yes, because they are connected to the Golgi. They are coming from the Golgi. So anything that is a part of this sequence of production, modification, packaging is a part of the endomembrane system. It's like a factory. It's like a production line. It's a conveyor belt. Those membrane-bound organelles which are not a part of this chain are outside of the endomembrane system. Who is not a part of the endomembrane system? Mitochondria and chloroplast, plastids. Mitochondria and plastids are not a part of the endomembrane system. Now look at the options. Anything with mitochondria has to be eliminated. Except the right choice, everything has mitochondria eliminated. Can we include ribosomes as a part of the endomembrane system? Ribosomes? Ribosomes, are they membrane bound? No. So endomembrane system is a system of membrane bound organelles which are a part of this entire production line. Okay, next. Matching are good. Same question. Same question every year. Aspergillus citric acid, Acidobacter acetic acid. Then Clostridium botulinum, butyric acid, Lactobacillus lactic acid. For effective treatment of a disease, early diagnosis and understanding of its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? ELISA. ELISA is enzyme linked immunosorbent assay. What do we identify with ELISA? Do we identify antigens or antibodies? No, what do we identify? Is it used for the identification of antibodies in the sample or antigen in the sample? Antibodies in the sample? Why not antigen? Both. See, ELISA is based on antigen-antibody reactions. ELISA is based on antigen-antibody reaction. Now, if you can design ELISA to detect antigen by having antibody in the test reagent, you can design ELISA for detecting antibody by having antigen in the test reagent. Alright, you can detect both antigen and antibody. Southern blotting technique is for DNA identification. What do you use for RNA identification? Northern blotting. What do you use for protein identification? Western blotting. What is Eastern blotting used for? Do we have Eastern blotting? Do we have Eastern blotting? What is it used for? Huh? Even I am not able to. Okay, next one. Which of the following characteristics is incorrect with respect to cockroach? We are having questions related to digestive system of cockroach. And they are giving this exam to select students to do MBBS. And the syllabus for this exam does not have digestive system of humans. A ring of gastric CK is present at the junction of midgut and hindgut. Gastric CK in cockroach is for the purpose of producing enzymes. It is present at the junction of foregut and midgut and not midgut and hindgut. So that is a wrong statement. Hypopharynx lies within the cavity enclosed by the mouth parts, which is true. 7th to 9th sterna together form a genital pouch. Yes. 10th abdominal segment in both sexes bear a pair of anal cerci. True. 10th abdominal segment has something else in males. Anal style. Persons with AB blood group are called universal recipients because, don't look at the options, give me the answer. They do not produce antibodies against either A or B. Blood group related discussion, we have had enough discussion. Dobson units are used to measure the thickness of Dobson unit. It is ozone. Dobson unit is used for measuring the thickness of ozone layer. 1 Dobson unit is 0.01 millimeter or 1 Dobson unit is 10 micrometers. Statements, and you have to choose which statements are correct. Alright. Again, same. You have 5 statements. You have to find out which statements are correct. So, if you find a statement to be wrong, any option with that statement should be eliminated. If you find a statement to be right, options without that should be eliminated. Now let's see, first statement. Metagenesis is observed in helminths. Is it a right statement? Wrong statement. So anything with A should be eliminated. Option 2 and 3 should be eliminated. Now check between 1 and 4. What is common to 1 and 4? C and E are common to 1 and 4. Now you have to check whether B is right or D is right. That's all. B. Echinoderms are triploblastic and coelomate animals. Is it true?
Yes.
It is true. Just cross check with D. Comb plates present in ctenophores help in digestion. No, they help in locomotion. So state option 4 is correct. In a cross between a male and female, both heterozygous for sickle cell anemia gene, what percentage of the progeny will be deceased? Don't write it down, just tell me by your mental calculation. I always want you to write down when it comes to calculations related to genetics. But just think. Both are heterozygous. Dominant, recessive, dominant, recessive. Sickle cell anemia is recessive, autosomal recessive. So both of them have dominant, recessive, dominant, recessive. So it is like the F1 of monohybrid cross. What do we want? Both recessive. F2 with the recessive expression is 3:1. That no, in F2 the phenotypic ratio is 3:1, where 3 are expressing the dominant trait, 1 is expressing the recessive trait. 3:1. 1 out of 4 is 25%. No? You can write it down and calculate. If you did not understand how you arrived at 25%, just write it down and calculate. So, let's say capital S, small s, capital S, small s. Both of them are this. When you do a cross between this, 3:1 or 1:2:1 is the 1:2:1 is the genotype. This is capital S, this is capital S, this is small s, This is what will be deceased, no? This is what will be deceased. Out of the 4, 1 is probable to have deceased. The probability of disease manifesting is 1 out of 4, which is 25%. Which enzyme is responsible for the conversion of inactive fibrinogen to fibrin in the coagulation cascade? Who converts fibrinogen to fibrin? Thrombin. Is thrombin present in the active form in blood? Prothrombin will be the form. Who converts prothrombin to thrombin? Thrombokinase. Who produces thrombokinase? Who produces thrombokinase? Huh? Liver. Alright, next. What else is there? RNA and reninase. What is the primary role of renin? Converting angiotensinogen to angiotensin. Increases blood pressure. Epinephrine is produced in which part of the body? Adrenal gland. Which part of the adrenal gland? Adrenal medulla. Select the favorable conditions required for the formation of oxyhemoglobin at the alveoli. Formation of oxyhemoglobin means what? If oxyhemoglobin formation is favored, what does that tell about the conditions? What does it tell about oxygen demand? If oxyhemoglobin formation is favored, what will it tell you about oxygen demand? Oxygen demand is more. If oxygen demand is more, will you have more oxyhemoglobin formation or dissociation? Dissociation. Alright? Oxyhemoglobin is like a good citizen. Oxyhemoglobin is like a good citizen. Carboxyhemoglobin is like a bad citizen. What does a bad citizen do? Wants to accumulate all the resources for himself. Does not make it available to others, irrespective of what happens to the others. That is a bad citizen. What does a good citizen do? If you have the availability, or if you have access to resources, you will hold it, you will take it, you will keep it with you. But if somebody needs it, what will you do? You will give. Wealth in a good person's hand is beneficial to the society. Oxyhemoglobin is like this. You should become like oxyhemoglobin. You should become like hemoglobin with the affinity to wealth, like hemoglobin's affinity to oxygen. When it is available, take it, keep it with you. And when it is required for the welfare of the country, give it. Alright? So, oxyhemoglobin association and dissociation is represented in an oxyhemoglobin dissociation curve. Oxyhemoglobin dissociation curve. Just you have to see with related to hemoglobin, related to hemoglobin, you will have this questions, have the questions coming from the dissociation curve. This is PO2, this is percentage saturation of hemoglobin. Percentage saturation of hemoglobin. Now, if the partial pressure of oxygen is low, very low, what will be the saturation? Saturation will be low. As the partial pressure of oxygen keeps increasing, what will happen to saturation? Saturation also keeps increasing. When the partial pressure crosses a certain critical value, saturation increases significantly. It will pick up oxygen very strongly, and at some point it becomes a plateau. Why? Saturation becomes maximum. 95-98% saturation you have it, and why do you have to pick up further? You don't pick it up. Now, if I say the oxyhemoglobin dissociation curve is shifting to the right, and shifting to the left. What does this indicate? If it is a leftward shift, what does it mean? If it is a leftward shift, what does it mean? With lower partial pressures of oxygen, there is more saturation. Correct? Even with the lower partial pressure of oxygen, hemoglobin saturation is high. What signal does it give you? And if it is a rightward shift, what does it mean? Even with higher partial pressure of oxygen, saturation is not so high, it is low. So if the oxyhemoglobin curve shifts to the right, what is hemoglobin doing? Is it associating with oxygen or is it giving up oxygen? Is it taking oxygen or giving up oxygen? Giving up oxygen when it is a rightward shift. When it is a leftward shift, what it is? Picking up oxygen. Now think about yourself as a wealthy person, accumulating wealth or giving away wealth. Like this. Hemoglobin picking up oxygen, accumulating oxygen or giving up oxygen. When do you want to pick up wealth? When do you want to accumulate wealth? When will you build more wealth? When everybody is happy, there is no demand for you to give up your wealth. The country isn't. Lesser demand for your wealth to be distributed. When do you give up more? When do you give up more? When there is demand for that. So now tell me, if there is a higher level of hydrogen ion concentration in the blood, what will happen to oxyhemoglobin dissociation curve? Shifts to right. Why? High pH or high levels of hydrogen ion concentration means oxygen is low, oxygen availability is low, demand is more. Whenever there is more demand for oxygen in the body, oxyhemoglobin dissociation curve shifts to the right. When the demand for oxygen is low, it shifts to the left. Shifts to the left. So whenever questions come asking you to tell what happens to oxyhemoglobin dissociation curve in a particular situation, we will do this. If temperature is high, if the body temperature is high, what happens to oxyhemoglobin dissociation curve? Shift to right, because in higher temperature, high metabolism state will be there, more demand for oxygen. When you are running, when you are exercising and running, what happens to oxyhemoglobin dissociation curve? Right shift, because demand for oxygen is more. When you are doing meditation, when you are doing pranayama and meditation, shifts to left. That's all. So look at this question. Select the favorable conditions required for the formation of oxyhemoglobin at the alveoli. Formation of oxyhemoglobin, favoring formation of oxyhemoglobin means, what do you have to tell about the demand? Demand is less. Now look at the condition. High PO2 favors pickup of formation of oxyhemoglobin. Low PCO2. Yes. Less hydrogen ion. Yes. Lower temperature. Yes. That's all. That's the one.
Sorry, if in case of H plus concentration, pH is given then?
Acidic pH, oxyhemoglobin dissociation, because you have to maintain the blood slightly alkaline. Neutral to slightly alkaline is the blood pH. Acidosis is dangerous. If there is acidosis, if there is increase in acidity, if the pH is lesser than 7, oxyhemoglobin dissociation. Shift of the curve will be to the right. Shift of the curve is to the right. 180. Sphincter of OD is present outside the door. Alright, okay. Right. Okay. 20 minutes. Need all of you, whoever had to give your sound bites, have you given? I hope all of you will give it. See, if all of you get through and get into a medical college, this batch would have created history, and there will be more such interviews. You know why this is being done? The council is presenting about whatever you are doing and whatever we are doing here to the Chief Minister.
Oh.
Alright. With what you have learned in these many months and what you have developed, if you continue to sustain with that, continue to grow as individuals that you have been initiated into, you will definitely be a very successful person that people will look up. Okay? Whether a medical college for MBBS or BDS or whatever, that I don't know. But I am very hopeful that everybody will be able to stick to the process. Alright? But don't give up on the process. Your process ends when? This process ends when? On the last day of your life, which comes naturally. Alright.